Hello, guys. It's my pleasure to introduce today's invited speaker, and also he is the last invited speaker, the last invited lecturer for this semester. So it's pleasure to have here Vasily Ramensky. Vasily is probably most famous for his works on methods for prediction of deleterious human alleles. Probably, you know, when you ask somebody why Vasily Ramensky is famous, what he's famous for, everybody will tell you, oh, this is the guy who was working on prediction of deleterious human alleles, right? So currently, Vasily is head of the Genome and Medical Bioinformatics Laboratory at the National Medical Research Center for Therapy and Prevention Medicine in Moscow. And his interests are in understanding the functional effects of genome variation and genotype-phenotype relations. So it's a pleasure to welcome Vasily, and he's going to talk about variant annotation and effect prediction. So Vasily, thank you very much for coming here with this lecture. The floor is yours.
Okay. Thanks for the introduction. Let me begin. Well, let's keep the abstract of the this lecture. You've probably seen this before, and you've probably seen a picture like this. These are variants associated with a phenotype. In this case, this is human height. The variants are scattered across the whole genome. Each dot represents a variant. The y-axis reflects the association strength. And suppose that we were able to sequence each variant in every subject, basically. So what we observe here is a true causative association, not linkage-driven. How could we interpret the results? That is, understand what is a possible biological mechanism behind each association. There are at least two things to keep in mind. First, the genotype-phenotype association goes through many layers of organismal complexity, only few of which are shown here. In particular, genes produce transcripts produce proteins. Proteins interact with each other and other molecules. Environmental effects and non-protein coding genes and gene regulation are missing from this picture. Well, besides this, each gene and variation within this gene resides at a unique location in the genome, and these locations are not equivalent as shown here. On the contrary, they are quite different. So let us take a closer look at the genome locations. Well, however unexpected this may seem, almost 20 years since the first draft of human genome was published, there is no solid and final sequence available still. There are still gaps and uncertainties that are being gradually resolved in each successive genome version with so-called patches. These are fixed patches and novel patches and updates. And of course, an individual genomes may be different, so a single sequence of nucleotides is mostly a consensus, an average approximation. And a typical consensus human genome sequence is not unique, is not completely unique. That is, roughly 1/2 of it consists of repetitive elements. Repetitive elements of all sorts and all length scales. Genes are only 20% of the whole human genome, and protein coding part of genes are only few percent, these so-called exons. There is a big ongoing discussion regarding the biological function of the rest of the genome. Most likely it's involved in cell and tissue specific regulation and is by no means junk DNA, as it was called recently. This table gives more precise figures. Roughly 20,000 protein coding genes. Most are with more than one transcript due to alternative splicing. So 150,000 protein coding transcripts. The remaining 40,000 genes and somewhat 80,000 transcripts are pseudogenes or lost genes. Genes with yet unknown nature and specific immune system genes and non-coding RNA genes. The latter are very diverse and abundant and are basically everywhere in the genome. We know what each RNA class may be involved. We know what each RNA class a particular sequence may be belonging to. For example, regulation of other genes, but we certainly do not know function of most individual RNA genes. But this is even true for almost half of protein coding genes. Just look at the table. These are protein classes ordered by descending abundance. So the nucleotide acid binding proteins and specifically transcription factors are most abundant, and hydrolases, that is enzymes as well. Please spend some time at this slide afterwards. Right now, we may take a look at the bottom line. So there are almost 10,000 protein coding genes with a known function. Amazing, isn't it? And all these overlap so that almost each DNA position is covered by really many coding and non-coding transcripts. So any variant typically belongs to many genes. Please keep this in mind. If we zoom in at a typical coding gene, we discover the quite complicated structure, which includes intervening exons and introns, and so-called canonical splice sites, GT and AG, between them, and many objects around the transcription start site, TSS, in the 5' untranslated region. The gene is transcribed into the pre-mRNA sequence, which undergoes splicing, which removes introns and thus produces mature mRNA like this, which is translated to a protein from the start codon up to this stop codon here, from this to this. It is thus important that the length of mature mRNA is a multiple of 3, otherwise no coding frame or reading frame is maintained. Location of start and stop codons, of course, is critical from this point of view. So disruption of either of them by mutation may be damaging. The gene expression process is complicated and regulated by many genome elements. This figure shows one of them, the promoter, with multiple elements that mediate the transcription regulation, like transcription factor binding sites and TA box and other boxes, that is conservative sequence motifs. Promoter is the so-called cis regulatory element, in the sense that it is close to the regulated gene. There are more distal or trans-regulatory elements called enhancers, silencers or repressors, or insulators. And whereas transcription factors bind to promoters and enhancers, DNA forms loops and such complicated multi-protein complexes with looping DNA enable gene transcription. So we should keep in mind that the vitally important regions of a gene are in fact much larger than just the coding parts of this gene. Can we put all these in order? All these gene relevant, gene important regions, can we put them in order? The answer is yes, to some extent. Let's make one step back, zoom in again, and take a close look at a coding gene's exon. A nucleotide change like this in a codon, like CAG to CAA, may or may not change the coded amino acid. Here it doesn't change. CAG changes to CAA. The amino acid is the same, or it may change the underlying amino acid from CAA to CAC. It is Q to H to histidine change. Or it may introduce a stop codon within the coding exon, like TGT changes to TGA. This is stop codon, so we introduce stop gain or nonsense, or this is a nonsense mutation. The molecular or physiological or clinical consequences of these three changes may be quite different. It is traditionally believed that, with some exceptions, the stop gain is most damaging, since it truncates the transcript. On the other hand, synonymous is most often benign or neutral, and missense, non-synonymous, has most wide range of effects from protein folding to no effect at all. And same is true for insertions and deletions in a gene sequences. Depending on their length, they either produce coding frame shift, like here, or just result in a short amino acid deletion or insertion. The former truncates the harboring protein, the latter may be quite diverse in its effect, just like missense variants. And it depends upon the length of deletion or insertion. If it's a multiple of 3, then it's fine. We're fine. The reading, the transcript reading frame is preserved, so we just insert or lose one or few amino acids, and otherwise the whole reading frame is damaged, and we observe the absolute dramatic change of protein sequence, and most likely many premature stop codons, like here. So we basically truncate or cut the transcript and the protein sequence. These are, by the way, examples of variant annotation. As we said before, it's a procedure that determines variant consequences for a gene protein based on its location relative to the gene sequence. So what I did here a moment ago, I was basically annotating the toy examples of variants, annotating them from the point of view of what they do to the transcript. And there are multiple tools for this, for variant annotation. Personally, I think the most powerful and comprehensive is the ensemble VEP, variant effect predictor, or VEP as we call it. It lists variant consequences virtually for all possible variant types, and assembles them in groups based on their impact. Here is the variant table with that VEP uses. Variant consequences. Here they are, and impacts. From high to moderate to low to modifier. Please see the details on the next slide. Now I think I can make a short break so that you can take a look at this slide. And these are groups of variant grouped by their impact, variance of various consequences, but grouped by the impact from high to moderate to low to modifier. So just as we said before, the high impact is for variants that disrupt or truncate transcripts completely, and moderate is for variants that change protein sequence with varying consequences. And variants with low impact are synonymous variants and splice region variants, that is, variants somewhere in the splicing region, but not affecting the canonical splice sites GT and G that we've seen before. And in fact, variants with low impact, like synonymous variants, may affect transcript splicing, and sometimes they do it, and there are some examples of this. But this is somewhat exotic, that is, this is somewhat rare, and most functional and clinical effect is for variants with high and moderate impact. And this slide shows a natural validation of the impact classification. It's based upon the ClinVar database. It's a clinical database of genetic variants seen in patients or their relatives. And variants in ClinVar are classified according to their disease causality. Pathogenic, that is disease causing. There are some rules to classify a variant as pathogenic or disease causing. Benign, that is not disease causing. There are many more of them, as you can see. And the most abundant group are the variants of unknown significance, VUS, for which we do not know, or for which there are conflicting interpretations of causality. This is the most abundant group. Just as one may expect, the first group, the pathogenic variants, are enriched with high impact variants, whereas such variants are mostly missing from the benign variants. Well, you see that there is virtually no red bar here, and the green is the largest one. And green and gray corresponding to low and modifier impact. So an unknown variants are the mixture of these two groups. Now, when a hope of some order appears, let me remind you that consequences are transcript dependent. So for a typical variant, you have to deal with all sorts of multiple consequences in a bunch of genes. VEP reports all, does not care anymore. So you have to make choices depending on your specific problem. For example, one may be interested in choosing a protein coding gene with reliable transcripts, and worst consequence in most representative and or reliable transcript of this specific gene. This is a practical problem. Well, just recently, in the course of one of our projects, my colleagues and I had to implement this reasoning in a simple tool for everyday use, so to say. That is, if if you have a bunch of variants, like 20,000, and in a bunch of genes, and you'd like to annotate them, what you typically get is much more than you need. You don't know how to deal with multiple mappings of a variant, like 10 mappings on average each variant. Now we know so many coding and mostly non-coding transcripts that each variant maps to a bunch of them, and your problem is to select one most informative, one most important. Apparently, this should be the one with the highest impact. But there is another player in the game, the transcript reliability. So probably you'd like to use the highest impact in the most reliably annotated transcript, or in the transcript of a gene that you know something about from biological point of view, that inevitably points to the protein coding genes, because what we know is mostly about protein coding genes. So you have to look at the protein coding genes first, to look at the best of them. I mean, the best from annotation, from validation point of view, the best transcripts, the confirmed transcripts. And in this transcript, we are interested in the highest impact, just like here in the table. So you build a non-redundant annotation for your variants. This is in some sense, this is not very complicated from a scientific point of view, but this is very important from practical point of view. And if you do this, you have all the treasures of variant annotation in your hand, but in the non-redundant manner. Now we will quickly review variant classes based on their impact. First, variants with high impact are also known as protein truncating variants, PDVs. Just as we said before, these are mutations to premature stop codons or nonsense variants, frame shift indels, and variants that disrupt canonical splice sites. Traditionally, all these variants are also called loss of function, or LOF, and this is still widely used. But now we know that not all PDVs, not all protein truncating variants are loss of function variants. Not all of them. Well, these are known protein truncating variants that are the most frequent cause of early onset Parkinson's disease. Early onset means 40 or 50 years. And they represent all known types of PTVs and knock out the PARK2 gene product. Well, you probably know that Parkinson's disease is the most common neurodegenerative disorder after Alzheimer's disease, and its prevalence in industrialized countries is up to 0.3%. And speaking with, speaking of variants with a moderate impact, most abundant in this class are the missenses, or non-synonymous variants. They actually give a first known example of a disease mutation. The so-called sickle cell mutation, it causes adhesion of hemoglobin molecules, and this in turn causes sickle cell anemia. And this mechanism reminds us that the proteins that are the target of missense variants are peptide polymers with very diverse spatial structures and very diverse features. Proteins are the building blocks of all, essentially of all cells and tissues. They make up all cells and tissues. And I give here just two of numerous examples of missense variants affecting their harboring proteins. Mutations that affect protein folding and stability. Well, for example, they introduce the charged residue into the hydrophobic core of protein, and the result is no soluble protein. It cannot be solved in water or blood or whatever. And another example is the site change in the hydrophobic core, which dramatically reduces the activity of the protein up down to 30% of the wild type. So on the organism level, this results in a disease called Porphyria cutanea tarda, whatever it is, well known and well described disease. And all this begins with a small missense mutation. Another example is the disruption of protein-protein interaction. In this case, this is von Hippel-Lindau syndrome, an inherited predisposition to a variety of cancer. And in this case, the missense variant destroys binding between two proteins. One of the proteins is not degraded. This results in overexpression of blood vessel or angiogenic growth factors, and this results in local proliferation of blood vessels around a growing tumor. So this is basically this variant has oncogenic effect. So this reminds us that a protein must fold properly and perform its functional interactions. So these guys target the most important protein features. There are multiple attempts to list and classify all possible missense variant effects in order to predict their effect. We now know that not all PDVs have detectable damaging effect, and this is even more true for variants with moderate effect, especially for missenses. Can we predict their effect? Yes, to some extent. We can collect multiple quantitative or categorical features of a substitution and its substituting site that is positioned in the protein, and in some cases of a protein itself, protein as a whole object, and build a supervised machine learning algorithm to predict its functional effect, be it damaging or functional or benign or neutral. And there are millions of methods for this. And the most effective feature mentioned 2 slides before, let me roll back, the conservation. This is a very wide notion. So the most effective feature that most of the methods use is position-specific conservation, or the analysis of orthologous proteins. The question is what amino acids are observed at this specific position, in this protein family. If the replacing amino acid is okay for any homologous protein, other homologous protein, then it's probably okay for the human protein. And well, the example here is there are 3 positions given here. We see that, well, for the first position, the arginines are rare, but other orthologs contain serine or threonine or other proteins as well. And if we are interested in arginine change to say serine or whatever, then we may expect that this change is most likely benign because it is seen in many other proteins, and apparently it has undergone millions of years of natural selection. And if it were detrimental or functional or damaging, it would not be fixed and would be purged by natural selection. So this is to some extent the site-specific conservation, not just the overall conservation of this position, but conservation that tells you basically what amino acids are okay at this position. And this approach has been confirmed and validated many times by all possible methods. Well, most likely the most straightforward is like this. If we apply the prediction methods to known variants observed either in patients here in the ClinVar database or in various human populations here in the exact database, now replaced by the GNOMED database, which accumulates sequencing results from literally hundreds of thousands of human genomes and exomes, and for virtually every variant in human population, it can exec or its ancestor. No, not ancestor, but its child GNOMED can tell you the it's this variant frequency in overall human population or in other human subpopulations. So for each variant you can check its frequency, and there are common variants with high frequencies and rare and ultra rare variants with very low frequencies. And the lower the frequency of a variant, the more likely it is that it may be damaging or deleterious or whatever you call it. That is, it may have adverse effects for the organism. And if it's rare, it may be new, or it may be old, but being under negative selection pressure and because of its functionality. So so we can check whether a prediction method correctly predicts most of disease-causing variants, and on the contrary predicts most common variants as benign, which is correct. The higher population frequency of a variant is, the smaller is the probability of it to be predicted as damaging. This is in agreement with natural selection and population genetics. So, speaking of variants with low and modifier effect, there are a few slides from now on. I think the few slides that report the recent developments in the field. For example, the splice IA algorithm that attempts to predict splicing from sequence. The substrate of this method are synonymous and intronic variants. We basically do not know much about them, except for the fact that in some cases they may affect splicing. And splice IA makes an attempt to predict their effect, and it is the method of a new era. It's based on big data and deep neural network algorithms. And well, of course, some people are very skeptical about these, but well, some people are enthusiastic, and well, I think the future will tell. I think I will leave these few slides for your homework, beginning with the previous slide number 32, slide number 33, 34, 35, 36. And maybe 37. These are relatively new methods aimed at prediction of non-coding variant effect. And this is a complicated thing because, well, any regulation is tissue specific. So tissue specific and cell specific. So you have to keep this in mind. And well, in the end, let me give two real-life examples from my colleagues and my papers. The first figure gives results of analysis of missenses and protein truncating variants in a relatively small group of important so-called haploinsufficient genes. There are from 2 to 3,000 such genes in the whole human genome. And haploinsufficient means that both copies of this gene should work, both should not be affected by any truncation or whatever may affect the gene. And these are most fragile, most vulnerable genes among all human genes. So you see that there are, roughly speaking, 10% of such genes among all protein coding genes. And of course, if we know that some genes are especially vulnerable, we would like to check if the mutations observed in a group of patients, for example, affect these specific genes. And this is a so-called meta-analysis of patients with 5 psychiatric diseases: bipolar disorder, schizophrenia, intensity deficit and hyperactivity disorder, autism, and intellectual disability. And we considered only rare and ultra-rare variants with frequencies below 0.1%. And the x-axis shows odds ratios for each one additional variant. And y-axis shows the number for each disease. It shows the number of methods that predict a variant as damaging. And this we see here that the more methods predict a variant as damaging, the higher odds ratio for the variant we may expect. And this is especially true for intellectual disability, where the genetic contribution may be the highest. We do know this from other studies, and from this point of view, we can expect these results. And these are ultra-rare variants seen. These are rare variants with a frequency below 0.1%, and these are ultra-rare variants seen in even fewer number of subjects. And these variants are the modern objects of study for statistical genetics. We look at the variants with expected most severe effect, with most rare variants, but we have to group them because each individual variant is very rare, so we can not do association studies on each individual variant, but we can group them together and consider them together. But to do this, we have to have a basis for such grouping, and this is where annotation and prediction methods excel. This is their area. We use them to group variants before we feed them into any statistical genetics association studies. And this is the area where the prediction and annotation methods currently are the most important, the most paying back. And the second example, these are the de novo mutations detected in Tourette syndrome. Tourette syndrome is a form of a complicated chronic tic, which has two components: the vocal tics, that is, tics involving someone's speech or voice, and motor tics involving involuntary movements that are almost impossible to control. And well, this is a mysterious neuropsychiatric disease, in the sense that we know that it is very heritable, that is, it occurs in families, and it's being researched for decades, and still we do not know the genes, or of course even more, we do not know specific variants that may predispose to this disease. So the Tourette syndrome is the area of very active research in the field of neurodevelopment and psychiatric disorders. And here we consider de novo mutations, and we consider de novo rates for mutations of different classes, and well, for some reasons, a new abbreviation is used here, LGD, likely gene disrupting. This is a new, I don't know why it was used, a new synonym for protein disrupting, protein truncating variants. And we see that in patients, the mutation rate ratio is clearly higher for the mutations of certain classes. These are LGDs, that is, protein truncating variants, and this is also true for missenses. And the highest effect is for those that we predict as functional. The ratio is 1.38. So and we do not observe this for synonymous and indels that do not change the reading frame. So this shows that annotation and correct variant effect prediction is quite important for statistical genetics, and specifically for rare variants for which we do not know, for which we have to apply specific statistical methods like grouping, and for this grouping we have to know what your variant is or maybe. So let me stop here and leave you with with summary of this lecture that summarizes some points that I was trying to deliver. Thank you for your attention. Well, okay, I hope I can be heard, and here is the question from Nadezhda. The question is, how frequently missense mutations may lead to disease? For instance, let's consider they are located not somewhere but in a protein domain, which is usually important for protein function. Yes, that's correct. What is the approximate probability that these mutations are harmful? Well, the answer is it depends. It depends upon what your specific substitution is. For example, I don't know if it's glycine to tryptophan change in the protein core. Well, most likely it would be damaging. It would be damaging for protein function, but it does not necessarily mean that it would be disease causing. That's a big and important thing. Not every function disruption is disease causing. There is a huge gap between damaging and disease causing, and well, we still are beginning to understand this. This was not clear some times ago, but well, it's better known now. On the other hand, if you change, I don't know, isoleucine to valine in the protein core, the probability of it to be damaging or disease causing is much lower, but it's still not zero. So basically what your prediction algorithm does, it answers your question for not just in general, but for every particular specific variant. And if you consider large groups of variants, like pathogenic or disease-causing variants in ClinVar, that is variants confirmed in patients and blah, 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 and so on, then the answer is yes from 80 to 90%, just like you've seen on the slide before. Thanks for your question. And if you consider common variants, then the segregating in a population with high population frequencies, that most likely that just a small fraction of them would be damaging, and most of good algorithms would predict this.
Can you hear me now?
Yes.
Oh, cool.
Perfect.
Right. Yes, sorry about that. So while so please ask your questions. So, and to give your time to put your questions in the chat, maybe I ask my question meanwhile. Well, first of all, thank you very for a very interesting book, and also for the introduction where you reminded a number of important things, which, you know, like, yeah, people tend to forget. I was specifically, you know, amazed to be reminded by the fact that for like 10,000 genes, we actually know very little about the function, because we tend to think when we read scientific literature, it looks like we know so much, but actually, we still have these huge gaps in our knowledge. So thanks for that.
Yes, I was amazed as well. Just like you, I was I don't know, I was looking at it again and again, and but well, what I had to do is to accept the fact that just 50% of genes have are annotated as unknown or whatever you call it. So amazing but true.
Yeah, and this is very important to remember about this, because like when you can identify the gene, right? Or like this, what is happening in GWAS, right? We identify regions and we have a number of genes to choose from. And it's important to remember that actually there is this 50% of genes we know nothing about, which we cannot even consider as candidates. But this doesn't mean that they are not really involved. So we are kind of used to consider only the proteins we really know about, right? But it's really important to remember that there is this, missing spot. We miss a lot in our knowledge.
Well, in any case, I would say that if your GWAS hit is somewhere near a protein, even an unknown protein coding gene, then you're lucky, because most of hits are in the middle of nowhere.
Exactly.
And you can only assume that that the closest gene, whatever it is, may be involved or maybe not, or maybe the variant is in a region that is involved in the regulation of something,
yes.
something, and so on and so on. So yeah, I don't know what is better, an unknown protein or absolutely anonymous region.
True. Yeah. I think the methods, but correct me if I'm wrong, but the methods you described, when we predict the functional consequences, we are kind of relying on the conservation. So we are relying that these guys having really deleterious effects. And then when we look into like standard GWAS, we expect that the GWAS, the variants identified via GWAS, they are actually under very weak selection. So in that sense, the methods you described are mostly tailored towards the de novo mutations and rare mutations of very big effect, which you expect to observe in Mendelian disorders, and less tailored to identify the variants which may be, involved into chronic late onset disease. Is my understanding correct?
Well, yes, to some extent. Well, basically, if you look at conservation, and we know that conservation works best, better than, say, to my big disappointment, and to my disappointment, works better than say structural features like I don't know, location and interaction. But yeah, so if we base our prediction on conservation, then essentially what we predict is the action of negative selection.
Yes.
But you basically you predict that based on its features, this variant is under negative selection. That it does not mean that it is disease causing, of course.
Yes.
And well, that's the reason for the existence of this gap that I mentioned before between disease causality and damaging, I don't know, and deleteriousness. Let me put it this way: there is a well understood now gap, but there are other things that are in my mind that I would be very happy to rehabilitate the structural stuff, the structural science, and somehow to get it back to business, because it's somehow neglected. And this is very sad, because we're just leaving a big piece of data, big piece of knowledge. And I think what I'm concerned with is to find a spot where the structural annotation, structural based prediction can augment, can work together with conservation based stuff. And I hope that we'll be able to find such areas to bring it back into practice. Say, well, I'd be happy to see the structural annotation where it is available, of course, to be used in clinical interpretation, if possible, and all kinds of things like this. By clinical interpretation, I mean when a situation when you have a disease and you do clinical sequencing and you find a possible disease-causing mutations, but you are not absolutely sure. And all sorts of evidence is used for this to make a decision for a clinical geneticist to make a conclusion and to write a report. And well, I hope that one day the structural based annotation for variants will be among the tools that are for everyday use.
Okay, so if I mean, I definitely taken only part of your message now, but just to be, sure if I understood you correctly, that if I'm thinking of interpretation for common GWAS susceptibility kind of variants, then you think that structural methods may be more relevant than those based on conservation, right?
No, I did not specify the GWAS stuff. Well, when I said this, I kept in mind maybe more Mendelian-like stuff.
Okay.
Well, for Mendelian diseases, we know that some structural features are very important, like disruption of protein stability. The other thing is that still we do not know how to predict this description computationally. We do not know how to predict, well, to some extent we do, but with limited reliability, how to predict protein stability change as a result of missense mutation. There are some good methods for this, but they're not perfect, so they should be used with caution. But what I did not mean the GWAS results specifically. There is an interesting thing. We know structural features for specific structural features for disease mutations, like they occur in protein core, they occur in the interaction hotspots, and so on. This is quite natural, so this is what you may expect. And the question that comes to my mind, on the contrary, the neutral variants that affect nothing are located at protein surface area. They just affect nothing. Neither hydrophobic core, no important interactions. So the question is, so these are two opposites. Mendelian disease variants and neutral variants, and apparently there is a whole spectrum in between, whole range of effects in between. And I'm interested in a specific question. To the best of my understanding, no one ever was interested. So the question is, what are these structural features of variants that are associated with, say, quantitative phenotypes?
Oh, yeah.
Those that we see in GWAS results. These are somewhere in between, of course, but I think they should have specific intermediate signatures. Like maybe they affect interactions more than stability, because stability is very fragile. if you disrupt it, then your protein is knocked out. So maybe they're shifted more towards interactions compared to Mendelian, well, and so on and so on. From this on, you can speculate just as much as you can. And this is a type of study that should marry, or I don't know, merge two basic science areas: quantitative genetics and statistical genetics, aimed at quantitative phenotypes, and maybe disease-related quantitative phenotypes, like, I don't know, cholesterol level, and structural bioinformatics, structural science, which basically deals with protein structures, I don't know, specific maybe substitutions, specific positions, and so on and so on. This, I think, well, this is very interesting. I hope that at some moment we can address this problem and see what is there.
I think this is, yeah, for me personally, that's very interesting problem. So I would be curious to see what will come out of your research. Maybe one last question, and this is, what do you think? Because there were like recent news that Google's DeepMind, because we start talking about this structural prediction, but there were recent news of Google DeepMind making a breakthrough. It was in Nature. I just sent the link to this. So what do you think? Is that a real breakthrough, or what is it? Because I'm like totally not specialist in the field, but they basically claim there is a, this Google DeepMind can now predict the 3D structures really well, and that's a big thing.
Well, yeah, I've seen a very active discussion on the Facebook, and well, some of my colleagues that are quite good experts in the field, they have their strong opinions. Or maybe, well, my opinion is not so strong. I just very agnostic. Like, let's see. Like any company, like Google, probably they're very close in that they do not disclose their method. They do not disclose the details. So we just have to believe that this is a breakthrough. Yes, that is true that they made a huge progress in one of the specific competition, the so-called CASP competition, targeted at prediction of protein fold by sequence. But, well, this is a very, very specific targeted competition, like one of the specific sports, like, I don't know, swimming or whatever. And we'll see whether it works for other areas first, and we'll see what is the resolution of their results. Like, if you can predict the fold in general, this is of course better than nothing, but this may be not applicable for some specific and multiple problems. Like, I don't know, like drug development, where you have to know your structure in detail to be able to model your interaction with a small molecule inhibitor. Knowing the fold in general is not very helpful, I think. But of course, it's great. It's very promising. Well, something should happen now, and will happen soon.
All right. I see that there are no further questions, so I think, well, Vasily, thank you very much for coming, for giving this invited lecture, for a very interesting presentation and for answers.
Thanks for inviting me. You're always welcome.
All right. Thank you, and thanks everybody for joining, and well, see you next time. And thank you, Vasily, very much again.
Thanks. Bye-bye.
Bye-bye.